No.
Okay, perfect. So am I starting? I'm very happy to be here. Thanks for the invitation, Awa and Oshine. I'm very happy to be here to present my work. As Oshine said, like I'm currently working in Trinity College and in also UCD College with Professor Dan Bradley and Professor Daly. My work is mostly focusing on ancient zoonosis during Neolithic in Southeast Asia. So we'll start. Oh, okay. Perfect. Sorry. So just to start, I will just like give a very small overview of the Neolithic transition. So around more or less 10,000 years ago, we start to domesticate certain animals, so especially livestock animals. We are goat, cow, pigs, and sheep in Southeast Asia. And my lab mostly focusing on cattle, goat, and sheep. So like I like also this very cool map from Melinda Zeder, which like you can see like what goat domestication start potentially in the like Zagros Mountain there, like all this region of Southeast Asia with like different time, but more or less around 10,000 years ago. But what are basically the impact of this domestication on the animals and the human also? Because in fact, when we start to domesticate animal, we obviously start to get access to very interesting product like secondary product like meat and also milk, later cheese and a lot of things. But what happened also is that when we start to link, our destiny with those animals, we also start to get their pathogens. And in fact, inventing farming was definitely a good idea for some stuff, but we also got like many pathogens which are like from those animals. I really like this graph. It was published in 2014, and it's the number of parasites and diseases that we share with domesticated animal. Comparing with the time of domestication. So obviously dog is the oldest domesticated animal, and it's like the one with which the most parasites, and following by bovid, sweet, and some ungulate. So definitely potentially the fact that we start to like being closer with those animals was definitely a potential route for pathogens. But like in what way? Like from the humans to the animals, to the animals to the human? Like we don't really know actually. In fact, pathogens have also different infection routes. Were like opportunistic, anthroponotic, sapronotic, and zoonotic or anthropozootic. So we just present quickly those different type of pathogens. So because it's important to understand kind of the different way and routes of like infections. First, the opportunistic pathogen, they are like pathogen where like most of the times they're part of your body, and they can infect you if you like your immune system is like low, or like your defense system is like not in the best shape of their life. Like it's a case of a lot of dental infection, like mouse infection, also like gut infection, and yeah, they are important, but they were not like transmitted by animals or anything. Then you have also the sapronotic pathogens, who are like very important. Even nowadays, they are like some organisms that you can find in the environment, like glass, soil, metal, and who infect you directly from the environment. So tetanus from Clostridium tetani is like very well known, and I actually got myself I got like a glass cut recently, and that's my hand after after the cut. So it was definitely, I don't think it was tetanus, but like I got infection because of like a glass cut. So those pathogens are everywhere, and they are infect humans since like every time, but like we are mostly a dead end. So when we get infected, we don't like transmit, I mean, theory, those pathogens. Then we have the anthroponotic pathogens, who are coming from the Greek anthroponos and apparently man, which is man, and osos disease. So those are the pathogens you transmit, so between human to human or animal to animal, actually, if they are from the same species. Like very important, like example are the smallpox, for example, like one of the deadliest disease ever, or gonorrhea, for example, hepatitis B. So many viruses mostly, and they are like very bad for humanity. But the type of pathogen that I will introduce today, and the more interesting one for this subject of domestication, are definitely the zoonotic and actually anthropozoonotic pathogen also. Because so what's actually what are the zoonotic pathogen? So zoonosis is an infectious disease that just jump from a non-human animal to humans. And anthropozoonotis is the reverse. So it's basically human which is giving like a disease to animals. They are extremely important nowadays. So 73% of emerging infection diseases have zoonotic origin nowadays. A total of 60% of human pathogens are actually zoonotic. I'm pretty sure we have like very good example of them, like COVID-19, for example, which is a bit important. And actually 25% of those zoonoses come from domestic animals. Like just doing the math quickly, that means that 15% of our pathogen nowadays are directly coming from domestic animals, which is very big. It's very important. That does mean like 15% of like our pathogen are like coming from our domestic. So it's important. But we start like domesticate animal 10 more like livestock animals, sorry, more or less 10,000 years ago. And what we are doing in Dublin is like we are trying to investigate the presence and the evolution of those pathogens in the Neolithic livestock. We want to know if the livestock was already carrying those zoonotic pathogen, or it was like something happened later. So, like, yeah, we don't really know. And we also want to explore the role of early animal domestication in the emergence of the modern zoonosis and pathogens. So, like, obviously, we are looking at Neolithic livestock, but which is also interesting for us is like, did those disease spread into like farmers where they are important back in the days and everything. So, but how actually we are doing that because it's very nice to talk, but like how can we do that? So what we are doing in Dublin is like we are looking at ancient livestock remains, so from Southeast Asia to find ancient so pathogen DNA. We are looking at ancient DNA of the pathogens. But how we do that, then what's the point to look at ancient DNA? So what we can do with ancient DNA is then compare the ancient Neolithic pathogens genome with like more recent or like completely like modern genomes to understand the evolution of this pathogen, the time of speciation, so we can understand when the pathogen start to spread and to diversify, all those things. So as I said, we are looking at ancient livestock pathogen DNA, but how are we actually getting this ancient DNA? So, like, just that's a very simple workflow overview of like what we are doing with ancient remain. So most of the time we are sampling bones, but like you will see also that sometimes we are sampling other kind of material. But most of the time we are micro-sampling the bone. So we are targeting a very tiny part of the bone. We are like most of the time trying to get the densest part because it's like potentially the one which is carrying the more ancient DNA. And then we reduce it into powder. But then like we take this powder and we extract it from, we extract the DNA from the bone powder, and then we build library and we sequence it. And then we do many bioinformatic analysis to find pathogens, but also to know like what species, so the host is present, like to check if like the bone is actually a goat and not a gazelle, for example, all those things. Yeah. So as I said, we mostly sample bones, but actually we found out that the diversity of material for ancient DNA sources is incredibly high, and also for pathogens. So that's the different bone I sample, and also some of my colleagues sample, like material in general. So we are targeting fetal remains. That's fetal bone, like goat fetal bone. We are targeting this kind of like remains because we are expecting that like potential fetus from like animal abortion would can carry any pathogens. We also obviously target teeth, petrous bone, which is the petrosal part of the head, which is there, and it's well known as more or less the best part of the body for ancient DNA. We also targeting long bones, coprolites. I never did that, but some of my colleagues, I'm quite happy I never did that. That's a bog butter, so basically where the butter was made. So potentially if you have like pathogen in the milk or something like this. Parchment, we recently discovered that you can find skin pathogen in some parchment. Also nodules. So sometimes you can, when you get sick, some pathogens are creating, are making calcifying nodule in your body. For example, Tenia can do that, some Brucella or something. And that's a very good source of ancient DNA pathogen because most of the time it's mostly ancient DNA pathogen, but it's pretty hard to find them. And also mummy sample. Yeah, that's a lot of different sources. But now I will present a bit how we actually do this, all this like ancient DNA work. So as I said, it's always a fashion week dumpster, and we are dressing up with those crazy big suits. It's like very warm when you walk with. And so it's covering all your body because you really don't want to contaminate your sample with like modern DNA from yourself and also from other bones. So we are like working with those very big suits. We are like cleaning every time, everything is bleached with ethanol, and we are trying our best to not contaminate the hood. Also, you see, we are putting thin foil that we are like cleaning. We are like also throwing it away all the time when we change samples. So it's pretty intense, and we are like really targeting we are just trying to not contaminate our sample. Also, like I present many, obviously, source for ancient DNA, but the best for when we are looking at ancient pathogen DNA are the teeth. I will explain what. So, because the teeth during the life of the individual are highly vascularized, so any pathogens in blood are like potentially detectable in the inner like root of the teeth. Like for example, Yersinia pestis, when you have Yersinia pestis, which is the plague agent, when you have a systematic infection is in your blood, you can easily then detect it in the dental pulp or yeah. So we are most of the time what we are trying to do, we are cutting the teeth into like that's a sheep teeth. And we are like then extracting the dental pulp and no, not the dentin, so just dental pulp to get, because it should be the more vascularized part of the teeth. And as I said before, nodules are also a very good source of pathogen. So if we can have like nodules and teeth, that's really the best for pathogen in theory. And also here you see that's a very dirty chunk of calculus. So dental calculus could be also a very good source of sorry, very good source of oral informations about the oral microbiome. But also you can get some oral pathogens. So it's also a very good type of material. So now I will present a bit of what we get, what we are extracting the DNA of the sample, because it would be amazing if like we were just extracting the bone, the bone, like we're just taking the bone, we making powder, extracting, and we get like, oh, only the pathogen DNA or only the DNA of the host. But it's absolutely not that. So what we get is like what I love to call soup. So it's the metagenome soup. So all the DNA, the metagenome is basically all the DNA in the sample. And what we are extracting is in all bones is obviously not only ancient DNA, because the sample, as you know, like can spend like thousands of years then in soil, the ground, so it could be contaminated with like soil bacteria, plant DNA, fungi, and yeah, it's very like obviously like you have life there, so yeah. And so if you're very lucky, like you get like from 1 to let's say 20% of the total DNA, which is part of the host. So when we are like looking at animals, sheep, goat, cattle, you're very happy with like 5% of DNA. And then a very tiny part of this total DNA, metagenome DNA, is then the pathogen aDNA. So it would be like less than 0.1%. I mean, if I get like 0.1% of the total is like ancient pathogen DNA, I'm super happy. It's like most of the time it's less than that. So it's very difficult to find ancient pathogen DNA because it's like a needle in a haystack. How to actually find our ancient DNA in this metagenome soup, because we have different approach, and the two main approaches are like broad metagenome screening and capture. So I will first present the capture, and then I will talk about metagenomes, because what I mostly do. So a genome capture is like using, I can explain that like very nicely. So you have RNA probes, and use so the RNA probes is like then designed to fit the ancient pathogen DNA. So you're throwing the probes into your DNA, and it should capture it. So it's like using targets to take the ancient pathogen DNA, and then you can recover the genome. But the thing is that, like for doing that, you need to know what kind of potential pathogen you have in your sample, which is not always the case. You can like just throw probes into a sample that you don't know, and actually this sample doesn't have this pathogen DNA. So for like making first sure that we have a potential pathogen, we mostly do metagenome screening. So we take the soup I present here, and then we use different tools to like take the DNA and check the taxonomy. If you have this pathogen, or if you have this microbial DNA from environmental DNA or something like this. It's pretty hard because it's very easy to have like false positive, but like for that, you have like different approaches that I'm presenting here. So you have different tools for doing that. Like some are like aligned based approach. So you align your DNA on different references and database. It's pretty slow sometimes, but it's quite efficient. So you have like relative reference with mapping, MALT, HOPS, competitive alignment, HSTAG, and so eMeta, a pipeline which is doing kind of all type of screening. Then also you have like a marker site approach. So it's like using some genes who are like present in pathogen DNA, and you know that like only those the species have these specific genes, and then you classify your DNA with this approach. It's good, but like it doesn't work super well on like pathogen. It's very good for like microbial community. Then also you have the camera approach, is like the one I'm using most of the time. It's using small DNA to small DNA, which not small DNA, small part of genome which are called camera, and to classify like quickly the reads from the DNA that you get. But then when we more or less know like what kind of pathogen we have in your sample, what we want to know is like it is actually really ancient, it is potential modern contamination, or like a close relative from the ground soil, which is like looks like this ancient pathogen. So we need for that to check if the DNA is actually ancient. So ancient DNA is really degraded. It's like shorter than modern DNA. That's already a very important stuff. Also, you have deamination reaction with time. So cytosine becomes uracil, and then the sequencer read it as T instead of C. So more C2C mean potentially ancient. So it's what you see here, like frequency of like C2C, the deamination. And also, yeah, for ancient DNA size actually matter because you really need small reads. Like if you have this kind of um, distribution, that means potentially your sample is like your DNA is ancient because you have like more like smaller reads. If you have like a bump here, for example, that's potential contamination. So if you really lucky and if you like have like all those parameters, so for example, you got like you find the good pathogen and then you have the good size, so potentially you have ancient pathogen there and you can potentially then capture it. So as I say, like I present first like all the techniques used for like getting ancient DNA. And now we just present all the bones and material I sampled during my first year of my PhD now. So most of my sample are actually coming from obviously the Southwest Asia and mostly from the Zagros region, and they are from the Neolithic. But I also sampled some from the Caucasus, some from like the steppe region, and one also from France. Most of my sample are obviously teeth, but also I have I sampled some petrus, long bones. Most of my long bones were actually fetal bones who didn't really work well. One nodule, the nodule was actually coming from France, and for a total 273 bones. So a big number of like bones, but like unfortunately a very important number of them didn't really work well because they're very old from like hot region, which is not the best for Asian DNA. But I got lucky enough to find some DNA, ancient pathogen DNA in some of my sample that I will present now. So what did we find? As I said before, I use a camera approach using Dragon Unique to screen all the DNA in all my sample. But like that's basically the result, the total result of like all my sample plotted and passing some thresholds of screening. Screening. Sorry, I'm losing the word. Detection. But as you see, it's pretty messy. You have like so many positive hits. Potentially, some are like many false positives. Some could be also opportunistic pathogens. So what we want to do is like zoom in a little bit and like cleaning for like important pathogen. So I just decided to present very quickly three of like the main results. Three results I got from all my sample. So one sample I was able to detect one positive hit for Brucella melitensis, which is a pathogen I will talk later. It's a zoonotic pathogen. For three samples, I was lucky enough to get to find Clostridium septicum, which is a saprotonic pathogen. So a pathogen which is we could contaminate animals from like ground or something. And also I found 5 samples with sheeppox virus, which is a very important sheep pathogen even nowadays. So now I will talk about a bit about like the Brucella melitensis finding because it's it's like a very important zoonosis nowadays. In fact, brucellosis nowadays is very prevalent, is the more prevalent bacterial zoonosis nowadays. So it's caused by the genus Brucella. So it's a bacteria from the genus, different bacteria from the genus Brucella. So you have different species who are infecting human, but also different animal hosts, like Brucella abortus, which is infecting cattle, Brucella melitensis, which is infecting goats and sheep, also Brucella suis, for example, which is infecting pigs, and Brucella canis, infecting dogs. They are like very common even nowadays in regions with extensive livestock farming. They are infecting people who are dependent of their animals. So it's very problematic in region when people like will eat raw meat or like even raw milk, but also you can get infected by fetus because those sorry, those those bacteria actually are making abortion in livestock. So it's economical problem because you can lose animals, but it's also infecting humans and animals. So it's a very big problem. So I will present this potential Brucella melitensis that we found in a petrous bone from Menteşe, Oyuk. This bone was dated from around 8,000 years ago with carbon-14. In this petrous was provided by Dr. Lurigo. And so the site of Menteşe is a pottery Neolithic site, so located in the eastern Marmara region, which is quite interesting is that the sheep proportion in Menteşe increased substantially around 8,000 years ago. Just something that I forgot to say is that this petrous bone was coming from a sheep from this site, female sheep. But okay, I will just want to present a bit the Brucella genus before start to talk a bit more about the result we got. The Brucella genus is very specific. Like you have also those species like Ovis melitensis, Arbortus, and Suis were like very closely related. Like their DNA is very kind of similar, so it's pretty hard to tell when you get like a positive hit for pathogen to say if it's like Arbortus, melitensis, Brucella ovis, brucellosis. It's quite important because they are like all kind of all specialized. So we did different analysis to find out what if it was actual melitensis and not maybe Arbortus or something like this, or ovis, who can also infect sheep. So we calculate the NEA, which is average nucleotide identity calculation. You basically take the genome of the bacteria and you look at like the similarity. And we look at all strain from Menteşe and we compare it with Arbortus and melitensis. And the NEA, it's between our ancient strain and the modern Brucella melitensis and Brucella arbortus are like extremely high. So like 99.80, it's like super high. Like usually between two different species, you get like lowest amount. So that means that like our ancient strain is like super close potentially to Brucella arbortus and Brucella melitensis. So to find out which one it was, we create like a set of SNPs sites who are like sites on the genomes of like different modern bacteria, and we try to find and we compare so this number of like those specific sites, which site we find in our ancient genome. So like what we found was very interesting. So like those number of sites are the sites who are like present in Brucella melitensis and Brucella arbortus. And the ancient genome actually carry almost all the sites who are like present in Brucella melitensis and Brucella arbortus. And only like 20, almost 40% of like sites were only specific to melitensis, but 0 from Brucella arbortus. So we were thinking that, okay, it's a Brucella melitensis, but it's a Brucella melitensis who potentially just split from like the common ancestor of Brucella melitensis and Brucella arbortus quite like, it was like very like close to the split of the two species. So what we did to check that, we first had a look at the phylogeny, and we can see that the ancient Menteşe is actually falling. It's actually basal of all the modern Brucella melitensis, but it's also very close to the split between Brucella melitensis and Brucella arbortus. That means that we know that this bacteria was more or less like 8,000 years ago old. And that means that potentially the split between Brucella arbortus, so the speciation between the two species, happened not very far away from the age of Menteşe. So for answering this question, because that's very interesting, if like the Brucella abortus, which is infecting cattle, and Brucella melitensis, which is infecting sheep and goat, just split during domestication time, is very interesting to understand why and how. And so to answer this question, we create what we call a time tree. So it's basically a molecular clock. So you use an ancient genome, and you compare your ancient genome with the modern genome and an outgroup. So in this case, Brucella abortus, and that gives you the time where like the species split and diverge. It's so much better when you have like ancient genome to help the tree to find the right date, because it's a molecular clock and sometimes it's not super accurate. So using our Brucella melitensis, Neolithic Brucella melitensis, we found that Brucella melitensis, the modern, and Brucella abortus split in more or less around 9,800 years BP, which is coincide with the rise of multi-species herding in Southwest Asia. That means that these bacteria specialize and start to split between two different species when the animal will start bringing together, like especially sheep, goats, and cattle. And that's very interesting. And what we did also, we wanted to understand a bit more about the biology of these bacteria, understand how these like splits happen and everything. So we start to look at what we call pseudogenes in the ancient genomes. And so I will explain quickly what is a pseudogene. So a pseudogene are defined as defective nucleic acid sequence related to functional genes. So in bacteria, pseudogenes are defined as genes that have been silenced by one or more deleterious mutation. Actually, what a pseudogene is a gene which is not functional. In bacteria, it's a very common way of evolution and some type of host specialization. And also some bacteria get like more pathogenic when they start to lose function because, for example, then they are not able anymore to live in the environment, so they have to infect a host or something like this. I want to show you also the impact of pseudogenization on the host specificity. That's a paper from 2020 on Salmonella enterica. And that's in red, the ancient strain were found in human. And that's also some modern Salmonella enterica who are like not host specialized. And that's a number of pseudogenes. And when you look at the Arbutus ovis and Typhus suis, and I think Arbutus equi also were like host specific. I think it's like pigs and sheep or something like this. And they are like all host specific. They have a number of pseudogenes which is so much higher than the more the generalist infectious one. So we wanted to see if the Brucella, ancient Brucella had the same pattern of pseudogene, or if they had like pseudogene comparing to modern and ancient. So what we did, we compare all the modern Brucella melitensis and the ancient Brucella melitensis with all the modern other Brucella to see what kind of pseudogene were specific to Brucella melitensis, so the species, and to see if the ancient strain had already those pseudogenes. So we found out that a total of six pseudogenes were fixed in Brucella melitensis, modern Brucella melitensis, against all the other like Brucella species, and our ancient strain like three specific pseudogenes who was already fixed around 8,000 years ago, and that three other pseudogenes happened after the 8,000 years ago, because our ancient strain didn't have it. And that was very interesting. I will explain why. Sorry. So before like explaining, I just want to show the mode of pathogenesis and of Brucella melitensis. So when animal, so mostly goat or sheep, get infected, then the Brucella go into the macrophage and stay in the macrophage to escape the immune system, and then most of the time, go into the placenta and replicate there. And so, as I show here, we had like three genes who were like already present, three pseudogenes, sorry, were already present in the ancient Brucella. And one specific one, which is called nosid, is implicated in the denitrification cascade. And the denitrification cascade for bacteria is used most of the time, sometimes, to do anaerobic respiration. And anaerobic respiration for bacteria, it's used for doing intracellular replication. And the modern Brucella melitensis show reduced capacity for anoxic growth, which is intracellular growth and replication to other Brucella species. And this reduced ability for intracellular replication might, proportionally increase the extracellular replication, or could be the result of like extracellular replication, but where? And something very interesting with Brucella species is that they have tropism for a sugar, which is called erythritol, so it's pretty hard to pronounce. And it's been shown that in mouse that Brucella melitensis, or like Brucella species, are really like here the red is like the presence of erythritol, and the Brucella pathogen are going directly there, even if it's not in the placenta, to replicate. So the Brucella have like a very high tropism for this specific sugar, and this specific sugar is found in very high concentration in the placenta of like ruminants, so sheep, and especially sheep and goat. Yeah, so I want to explain why this nosy pseudogene is very interesting in terms of like evolution. It's because like the ancient Brucella melitensis, so before 8,000 years ago, were already infecting the animals in that way with a high tropism for erythritol. And that means like before they had a functional nosid, but now, but after that they had the pseudogenization, so a non-functional one. And that showed that the Brucella melitensis was already infecting a lot of animals. And also this pseudogenization make like it's then more like it's like mandatory for the Brucella melitensis to infect the placenta to replicate and then spread via like abortion and fetus. So definitely this pseudogenization saw also adaptation and increase the pathogenesis of Brucella melitensis, and that's happened before 8,000 years ago. So that was fixed very early in the Brucella melitensis. So that means it's like potentially a very important one for the evolution of this pathogen. So that's a potential scenario of like the evolution of Brucella melitensis. That's just a scenario, but it's like potentially what could happen. So like during domestication, like especially at the time of like the split between Brucella abortus and Brucella melitensis, so like multi-species were like start to bringing together, and we can potentially think that a more generalist Brucella animal was able to infect both animal or cattle and sheep and goat. And then because of the spread and the diversification, because of domestication and the fact that the animals were keeping together, the Brucella start to spread between animals and then to specialize through those like different species like goats, sheep, cattle, etc. and then also infect humans, because like Brucella abortus and Brucella melitensis both infect humans. Yeah. So just some takeaway about this Brucella work. So the ancient genome highlights some no-zip pseudogenization, which is linked potentially with erythritol preference. The timing of speciation between Brucella melitensis and Brucella abortus is around 9,000 years ago. And that's coincided with the rise of livestock herding in Southeast Asia. So suggesting that domestication definitely influenced pathogen evolution. And what are the implications for Neolithic farmers and also for the livestock? So we can expect that close contact with livestock during the Neolithic for the farmers likely facilitate the emergence or spread of zoonotic disease through human. So definitely affecting the animals and the Neolithic farmers. So these works have been recently published in Nature Communication. If you want to have a look, definitely go. So nice paper, I hope so. And yeah, just checking. Okay. I would like to acknowledge all the people who like work with me in my lab in Dublin. So all my colleagues here, also my supervisor, and like all the person in my group, which is me and my supervisor, and also all my collaborators, and also SFI for the foundings. And thank you. And if you have questions, go ahead. Thank you.